So first of all, Klaus, I would like to thank you for the invitation to speak at this AI Meet conference, or the first Nordic AI conference for young researchers. I think it is inspiring for me to be here because we both come from two fields that are developing at an accelerating rate at a very, very high speed. Both neuroscience and AI sciences do that. Both are young fields, or at least fields that have grown tremendously during the past years. And I think the more we learn, the more we realize how much we can actually contribute to each other between brain sciences on one hand and AI on the other. And it's particularly exciting then to see how many young researchers are present at this conference, and that you have the opportunity to meet with each other, to discuss with each other, and to form connections that may last for life. So arranging this conference is a great idea, and I hope that my talk will inspire some of you perhaps to take up the connection to the brain sciences. What I want to do in my talk is to show you some of the fundamentals of how the brain works, some general principles, also will tell you how much we have learned recently about higher brain functions. That means intellectual activity, about human intelligence, and also show you how much we don't know. And I will also spend some time at the end of my talk to try to make some obvious comparisons between brains and computers to see where we can connect in the best way. So the human brain has almost 100, at least 80 to 90 billion neurons. And these neurons, these are connected to each other. So you see they have very, very long processes and go very far away, but in the end they connect to other cells. And the number of connections is very variable between these neurons, but an estimate is approximately 10,000 on average between neurons. Now we know that the information in the brain is particularly located in these connections. We call them synapses. That's the areas where brain cells or neurons meet each other. But this is so complex, and these synapses are so small that we can't really observe them when humans or animals are performing tasks. So it has been considered quite inaccessible for a long time. However, at the same time, we know that this is the material of human intelligence. This is what intelligence, or perception, memory, language, thinking, planning, decision-making, all that is made of. There is a very, very strong desire to actually understand how these neurons work together and how information is encoded and stored and retrieved and used in the brain. So this has become possible during the past decades, and particularly during the recent years, when we actually have the tools to infer the activity of the right neurons in the right numbers and at the right locations. But how do we do that? And then I think it's important to just take a step back and ask, how would we go about? Would we begin with the human brain with its approximately 86 billion neurons and not so easily accessible as you can imagine? Well, very much of the principles of brain function are preserved way back in evolution. And what you see here, starting from the right with the human brain, is a number of neurons in brains of different species. So then as you go through monkeys to rat to the mouse, you're down in the mouse to approximately 71 million. And if you go even further to the fruit fly, for example, you're down to 200,000 only. And if you go even further to the nematode C. elegans, you're down to 302 neurons. So you can go all the way down to fairly simple networks of neurons, and then depending on how advanced the functions you are interested in, you can actually study them at a number of levels. And much of what a brain does is actually present even in the simplest organisms. It's just like DNA. you don't need to study humans to understand how DNA works, because it's so fundamental. It came so early in evolution. So what we do in neuroscience is we try to go as far to the left as it is possible for the particular case. And the reason for doing that is, of course, evolution. So we all come from the same ancestor. And what you see to the left is just a cartoon of mammalian evolution, where you then at the bottom have our common ancestor, and we have our cells at the top right, and then we have the branches. But very, very much is common across mammalian brains, and for that reason we can actually learn a lot by, for example, studying rats and particularly mice. So this just shows that even if the brains look very different, I mean certainly the size is different, actually the way they are organized in mammals is quite similar. So this shows the cortex. That's a sheet that covers most of our brain, and which is thought to be particularly involved in all kinds of intellectual activity. So you can see that this is how neurons are organized across layers of the cortex in humans, in other primates, in carnivores and rodents and so on. And you just see the similarity, right? So I mean, if you are looking for the fundamental principles, no need to really go to the humans. And also this applies to how the neurons operate. How do they work? So the fundamental unit of communication in the nervous system is the same across all species. They all communicate via a combination of electrical signals, which are the signals that pass within neurons, and then chemical signals that go between neurons. When the signal is passed onto a synapse, then chemicals, neurotransmitters, are released and then go over to the next neuron, and then you get an electrical signal again. So this is fundamental, and this applies to every species. And within neurons, the fundamental unit is the action potential, as we call it. And this is a binary form of communication. It either is on or it's off. And this was discovered a long time ago, but the ones who actually understood it and found the mechanism was Hodgkin and Huxley in the early 1950s. They worked on squids because they have very, very large neurons, so it was easy in those days actually to study them. And what they found is that this unit, where there is a change in the electrical potential of the neuron, goes from minus 70, for example, millivolt to plus 40, 50. And this is a sudden change where it's either at a low hyperpolarized level or a high depolarized level. And you can see the curve there. It takes about a millisecond. That is caused by currents of sodium and potassium that are in a certain sequence. So this unit then is fundamental, and it already is something I will come back to, that the binary type of communication within neurons is one of the things that you might at least superficially compare to a computer. But there are many caveats about that, and we'll come back to that. But let's now focus on space. And why space? With space, I mean, where are we? How do I know where I am? How do I know how I get from here and if I want to escape, how I get out the door, for example? This is information that it is a high-level function. It is not related to the sensory systems directly, because it depends on brain areas that are far into the brain, far into the cortex. And even so, even if it is so high level that it has been difficult to understand it, there is actually quite a lot of progress in this area, and we do know the basic nuts and bolts of the space system in the brain. And that's why I want to tell you about it, and also why we do much of our research on it, because we know more about space than most other cognitive functions like planning, thinking, which is way more difficult. This is then the reason for studying spatial orientation, and it is, as I said, one of the best understood functions. So where should we look? Well, this is a human brain again, and you see two areas highlighted here. Both are in the medial temporal lobe, which is kind of behind your ears. It's the hippocampus, and it is the entorhinal cortex in red and in blue, respectively. So they are both on the inside of the cortex, so that's very difficult to show on the cartoon, but it's actually as the cortex folds underneath and goes back on the inside. But anyway, just behind your ears you have these two structures, which are important both for knowing where you are, but also very important for memories, which I will come back to. But studying these brain areas in humans is not an easy thing to do. Of course, you can study these brain areas in patients who have damages to these brain areas, or you can do functional imaging studies where you put people in a scanner and then watch the activity of these areas. But it doesn't have the resolution you need to understand the computations. To understand the computations, you need to have the resolution of individual neurons. And that you can even do in humans, because there are some humans who have very, very small electrodes implanted into their brains. And the reason is that these are patients with severe epilepsy who need these electrodes to actually find where the epilepsy starts. But there are obviously not many of those around. So if you want to do research, it is what we do is to do that in rats, and nowadays much more in mice. And this shows an illustration of this. I now want to take you back to 50 years in time, to 1971, where John O'Keefe, with whom we shared the prize in 2014, what he did was that he recorded activity from the hippocampus. You remember the red area in the human brain, but in rats. The rats were running around in a box, like the one you see there. And this one is, of course, not to scale, because the box is 1 by 1 meter, typically. And then he, for the occasion, then plugged in a connection to very, very thin electrodes, or actually sensors that were in the hippocampus, so that he could pick up electrical signals or action potentials from individual cells in the hippocampus, and then sent this to an oscilloscope and stored it on a computer. So what did he observe when he listened to the activity in the hippocampus? That is shown here, and I illustrate that with a movie. So what you will see is a rat that is walking around in a box. This is seen from above. The box is 1 by 1 meter. The experimenter is throwing out small pieces of chocolate all the time, just small crumbs of chocolate. And the rats like that, so they will run around all the time just to find the chocolate. And this is just a trick that we as experiments use to have the rat visit every possible place in this box. Why do we want that? That will become clear very soon now. I'm going to now start the movie, and you hear some noise. That is not noise, it is actually the activity of one single cell. So each time you hear a bop bop, then it is an action potential from that cell. And you'll also see it by red dots that will appear on the screen. Maybe. Good. So now you hear the activity of the neuron in real time. And you can see already now that this neuron in the hippocampus is active only when the rat is in the upper left corner of the box. When the rat is in other places, there's no activity in that neuron at all. So this neuron is active at a certain place in the box, and for that reason, O'Keefe, who found this in 1971, called these cells place cells. It can also be illustrated in color code here. So warm colors means high activity, blue means low activity. The same box seen from above, and you can see that the cell is active in the upper left part of the box. Many neurons were recorded by O'Keefe and colleagues in the hippocampus, and they found that each of them had a different area, so that they were active, so that in total every possible area in the box had a different combination of active cells. And for that reason, then O'Keefe and his colleague Lynn Nadell suggested in 1978 that the hippocampus, these place cells of the hippocampus, are actually a part of the brain's map of space. And such maps of space had been suggested already 30 years older by psychologists, but they didn't have access to the brain activity, so they couldn't really tell whether there were such maps or not. But here was the evidence. It is not a map in the strict sense that exists in other sensory areas of the brain, because neighboring cells could have different firing areas. But the idea was that if you or a computer had access to the activity of all the neurons in the hippocampus, you can tell exactly where the rat is at any given time based on the combination of which cells were active. But then this raised questions, because this is in the middle of the brain. It's far into the cortex, into the hippocampus. So how can such a map just arise from nothing when there's no signals that come from the outside that actually tell where you are? You have no space sensors on your body, right? So how can this come about? And that led to 10 years passed. No one knew where this came from. But then by accident, two people here, so Jim Rank and his student Jeff Taube, recorded, so it was Jim Rank first who recorded in a neighboring area of the brain. It was called, they called it postsubiculum, we call it dorsal presubiculum, doesn't matter for this context. But what they found there, and it was just when they had these electrodes down again, but they mishit, so it went into the wrong area, and suddenly they found cells that respond to not place, but direction. So which direction was the rat facing? Was it facing to the right, to the left, or forward, or backward? And you see an example of such a neuron here. So on the x-axis you have the head direction of the rat. On the y-axis you have the firing rate or the activity of that particular neuron, and you can see it's highly, highly selective for one particular direction. And then other cells responded to other directions, and again you could figure out from the combined activity of many neurons exactly which direction the animal was pointing its head. So direction and place obviously are probably part of the same navigation system. You need both to know where you are and you need to know the direction in the environment, like the map and the compass. So then another 15, 20 years passed actually, it came to 2005. And then here in Trondheim we recorded activity in an area that linked these two other areas in the middle, a kind of interface area called the entorhinal cortex, which is the blue one that you remember from the human brain. So again, rat is running around in a box, and we were recording activity from a single cell in this brain area. And the result, you'll get the result to the left there, but the result is also shown to the right. You will find a new type of cell that is active also in certain areas, just like the place cells of the hippocampus, but in many areas. And those areas are very tightly, they're like tight blobs, but they are many of them, and they form a very, very periodic, regular pattern across the space. And you can see the pattern. It's triangular, or actually equilateral triangles, or more correctly, a hexagon that repeats itself all the time, just like this. It is a grid that exists in the brain, and it was obvious that that kind of grid is maybe like a coordinate system that the animals could use to measure their distance and direction in the environment, which they couldn't get from the other types of cells. It illustrates a development that has taken place. Then since 2005, we also found a number of other types of cells that have very specialized functions in navigation. But what's common to them is that until a few years ago, both in this area of brain science and in other areas, most studies were done at the level of single cells. You found out what does a cell respond to. One cell responds to direction, another one responds to place, and so on. But we all know that's not like the brain works. The brain works in circuits, in networks. That means hundreds to thousands of neurons that work together to accomplish a basic function, almost like a CPU in a computer. So this is where we need to study brain function. So that hasn't been possible simply because of a lack of tools. But during the past 3-4 years, this has completely changed. And now there's been a technological revolution, partly actually started out with some major funding initiatives that came almost 10 years ago, including Obama's BRAIN initiative, which and there was also a European component, and now it's all over the world, which actually enabled the development of technologies that could pick up the activity not of 1 or 2 cells at the same time, but of many, many thousands. And I'll show you 2 of them. So here is a microscopy version, 2-photon microscopes, which allow activity of cells to be picked up live when a mouse is running around in a box. So you see again the same box, mouse is running around. The mouse is not bigger than this. It has a microscope that weighs only 2.4 grams, very, very small, and still gives the resolution that you previously got only by putting tissue under almost half-ton microscope that was several meters large. So this is a major advance, and it is a development that has taken place over 20 years, but the breakthrough came just very recently and is much due to Wei-Jan Zhong, who is actually in our lab now and has developed this so that we actually can use it for mice that run around freely. And just to show again, what you see here is in 4 different planes, the activity of cells in the visual cortex. You see them blinking on and off in parallel with the mouse running around. And it's about 1,000 cells that can be distinguished in this way. And how can you see them? That's because they genetically express calcium, fluorescent calcium, so that you can pick them up with a microscope. So you can actually watch live then how the activity changes in a neural network during performance of behavior. And this then gives rise to then possible, just show as an example, here are the cells and different colors refer to different types of cells, grid cells, head direction cells, and so on, and you can then map them out and understand how the neural circuits work. What I will do is I will focus now on a different technique, the second major advance of technology that has taken place and which also allows us to get thousands of cells. That is the electrical approach. So with the microscopes you can watch the activity, but it still has a limit on the temporal resolution, at least not better than several tens of milliseconds, whereas with the electrical probes you can get down to far beyond milliseconds. I'll show you one. You see them here on the. This is an example of such a probe. So what goes into the brain is the upper part that sticks out. So this is previously that was a wire that was uninsulated at the end, and you could pick up the electrical activity there. Now this wire is actually not a wire. It is a computer chip which has lots of spots, thousands of spots, where you can prick 384 of them for each recording, and then just listen to the activity in that spot, so that you can in total then cover a very large area and get many cells. And the most recent version now has 4 of these, like the one you see to the left, 4 of these next to each other, so that you can even further increase. And it's also miniaturized, so now it's so small that just weighing 1 gram, the whole thing, so that a mouse can easily carry this. And then you do this again while the rat is doing its thing, running in the box and picking up chocolate pieces. But this just shows an example now. So again, you remember the color map from the play cell when the rat was walking around in the environment. So this is an example recording from Rich Gardner in our lab, a postdoc in our lab, who then recorded 2,460 cells. And out of those, 483 were grid cells, and out of those, 149 were from the same subclass of grid cells. So each color maps for each of them are shown here. So light color means, yellow color means high activity, and blue means low activity in the box. What is the relationship between, how do they work together? So another way to display the activity there is to plot it across time. So on the x-axis in the right plot here, you have time. And then on the y-axis, you have cell number. So each line here is actually 149 lines. So black means active and white means not active. So you can see the different cells that all are the grid cells, when they're active, when they're not active. And then you can make a slice through time and then just ask for each time slot, which could be, for example, 100 milliseconds. And you can go through the entire event when the rat is running in the box and then make such time slices. So in principle, this activity in each time slice has a huge number of possible forms that the activity could take, right? Because 149 neurons, if you allow every possible combination of activity, you're working in a 149-dimensional space, which is difficult for us humans to handle, at least for the human brain. But you can break it down to the the primary dimensions, those that explain most of the variance. For example, like this is what we did then, still with Richard Gardner, but now in collaboration with also with people at the mathematics department, so Erik Hermansen, Ben Dunn, and Niels Boas, and also several others involved here. But if you break it down to the three most important dimensions by doing using standard techniques for dimensionality reduction, you actually get these point clouds that you see here. So each dot in this cloud is a segment of time, like the red bar there in this activity. So it shows activity plotted on those three most important dimensions. And what you see then is that this cloud actually forms a torus. There's a hole in the middle, so it's like a donut. And activity moves around on this torus, so you see this trace that corresponds to the activity when the rat is walking around in the box. And this happens both when the rat is walking in this square box, when it's walking on a kind of a track, or even when it's sleeping. So in all these cases this happens. But the problem with these dimensionality reduction techniques, where you look at the activity in the point clouds, is that it's not so easy to quantify it. If you really want to quantify the degree of torusness here, then you have to use other techniques, and I'll just quickly go through that because it's not really the main point here. But there are techniques from topology, and this is where we have the collaboration at the mathematics department, where you can actually count the number of holes in this n-dimensional space. And in this case, we counted the number of holes in 1 dimension and in 2 dimensions. So for a torus, which you see to the upper left there, you expect 2 rings in 1 dimension, 2 circles, and then you expect 1 hole in 2 dimension, which is the inside of the torus. And the lines there that you see in green are just examples of that. So this shows recordings from both when a rat is walking in a box, when he's running on a track, or when he is sleeping, either REM sleep, as we call it, as dream sleep, or slow wave sleep, which is a non-dream sleep. And in all cases, you see 2 holes in 1 dimension and 1 hole in 2 dimensions. So bottom line here is simply that regardless what these rats do, they have the same activity structure. That means that these grid cells do not only operate when the rat is running around in the box, they even operate when you're sleeping or resting or whatever. So, and they always move on this toroidal manifold. And now you may wonder, why do I tell you this? The reason is that this is an example that confirms major predictions from theories that exist in neuroscience. You can imagine that neuroscience has developed so much during the last years because we get so much data, but what is lagging behind is theory. How would you explain that these things happen? The observation of this torus actually confirms predictions from a major class of theories in neuroscience. They are called continuous attractor network models. So they've actually developed from statistical physics, but the idea is simply that when you have networks in the brain that are coupled in certain ways, and especially have strong connections between cells in the same circuit, then you can make the activity move around on a smooth surface in whatever plane you want. This has been suggested to underlie the brain's map for space, and this it was even suggested, as I will show you very, very soon, that this should be a torus. But let's then begin with a simpler case. And the simpler case, so if you think this is difficult, don't worry. It will take 3 slides and then it gets very, very easy. But I'll take you through these still, because I want to show you the importance of these, of having theoretical models of how things work, because it comes back to the neural networks that you may also be working on. So the idea here in these theoretical models is that neurons shown in color, the circles, are arranged in a ring, not in anatomical space, but you can imagine that they're arranged in a ring, and then that there are connections shown in red between cells that have similar properties. And these are the so-called head direction cells, those that are active only in a certain direction. So at the top in red, you have those that are active when you go in the up direction, and then at the bottom in dark blue, you have those that are active when you go in the down direction. So now there are strong connections between cells with similar direction tuning. That creates a bump of activity because they all excite each other, and then there's inhibition of the rest. So the fact that similar cells excite each other causes a bump of activity in this ring of neurons. Then there are inputs coming from the outside that then drive activity around in the ring. And to illustrate that, I will show this movie of this cat toy. The bump of activity is the ball, and then it can only move around in this circle. There's no other way it can move around, and it has to go continuously around, and it never jumps out of the circle. It has to stick there. And that's exactly how this bump of activity moves around in the head direction network too. And which cells are active at any given time is then dependent on where the bump of activity is. So this can then be moved to 2 dimensions. So this is 1 dimension because a ring is just 1 dimension, right? So this is a 2-dimensional version. Again, a bump of activity caused by cells that connect to very similar cells. So each circle here is now 1 place cell in the hippocampus that has its own area of activity. So the square is the box where that is moving, and then each ring is an area where a cell is active. And those that are active at the same place, in the middle in this case, excite each other. Then you get a bump of activity. Then from outside you get signals about speed and direction that the rat is moving, and that is then moving this bump of activity around in this brain map. So this is just theory, it's suggestions, nothing more. But then the idea was, if this is the case, and then this bump of activity comes to the end of the network, what happens then? So it's like the end of the world problem that we had about 500 years ago. The solution is that this can only work if you wrap it around, and it's actually circular. So if you wrap this around, and if the unit of wrapping is not this square there, but is a rhombus, then you actually get the hexagonal, you get this torus that you see at the bottom here. If you then move around on the torus, you actually get the hexagonal pattern, the space that the animal moves on. So this predicted that there has to be such a torus, and that's now what we have shown. So it confirms ideas from, or theories from, predictions from theories in computational neuroscience, as we call it. And this computational neuroscience is the field of neuroscience that I want to emphasize for you, because this is where probably artificial intelligence and neuroscience can meet in the most fruitful way, as I will come back to at the end. The same hippocampus and entorhinal cortex that is so important for space is also important for other critical functions like both time and memory. So this is a picture of a patient who died in 2008, Henry Molaison. He was operated for very serious epilepsy in 1953. And what the doctors didn't know then, they had to take away the hippocampus because that's where the epilepsy, the seizures started. And it was so serious that he would not survive if they didn't do it. But then they took away the hippocampus, and what happened was that then his memories also were gone. Not all memories, but all new memories. So essentially, he had a syndrome very similar to what you have in Alzheimer's disease, that you can't really form any new memories any longer, and it even has some effect back in time. This just shows his brain where the hippocampus was removed. You see some black dots quite far down there. But this was important because it started a half-century of work on the role of hippocampus in memory formation. And then one of the things that are lost in those memories is the memory of where you are, memory of space. Space is tightly integrated in this, And the reason, you can also see that just by the fact that what we call episodic memories, that's memories of any episode, anything that happens during the day, is always connected to space. So whatever you remember you did this morning, for example, before you came here, there's also an implicit memory of where you did this. There's always a space in it. That just shows how integrated space is into memory of that kind. So how do these memories work? Of course, I can't tell you that in 3 minutes, but there's still some basics I can communicate here. So again, we are talking about, at least in the hippocampus, millions of memories, perhaps billions, we don't know. It depends on how you define one memory. But in any case, memory has the challenge not only that you may forget them, but also that you need to keep them apart and not mix them up. And you can imagine that memories are stored in the connections between cells in a network, like the networks that you saw before here. And then just for illustration, imagine that the memory is stored in the connections between the black cells in this network. So the connections between those cells are strengthened beyond what they were before. That means that if you reactivate this memory or recall it, then you will bring up activity in these cells. But then there might be another memory where it was quite similar, for example, the memory of what you did not this morning, but yesterday morning. And that may be also stored in a network, but then slightly different. The hippocampus has to try then to get them as different, use different cells or even different connections. But of course, you can't use only different cells, because then you will use up the capacity very soon, even if you have millions of neurons in the hippocampus. So you need to rely on overlap and then have unique combinations. But still, making memories as different as possible is one of the things that this memory system has to do. And the other thing is to retrieve or bring up again, recall memories in the right way. Because when we recall a memory, we never start out exactly with what was encoded in the first place. You begin with something that is just a part of it. For example, imagine that as the 3 dots, green dots at the bottom. And then having enough information then makes the whole memory reactivate, and you get the others just activated as a consequence. And the reason why you can do this is that these green ones are so strongly connected together that if you just activate a little part of it, you can get the rest for free, and you perceive it as a memory. However, the problem is then that memory works then by just putting together pieces, and in this process where you reconstruct the memory, you also need to use the rest of the brain to make sense of it. And that is why when memories often may become false memories, because you try to make sense of it, the brain tries to make sense of it, and sometimes produces a memory that looks quite true, but isn't actually true, simply because it makes sense. It's coherent with the rest you know about the world. So that's memory in the human brain is definitely not like memory in a computer. So about memories, I also wanted to emphasize that memories are not just static, they occur over time. What is the role of time? How is time encoded in the brain? That's we have known quite little about that. It's far behind the area of space. But there's some work, and this shows some work we did ourselves about 3 years ago, where Albert Sau, a student in our group, recorded activity in the same box, but did that over approximately 10 trials after one another, with a few minutes in between each, so that the whole thing went on for 1 to 2 hours. And what he then found was that there was activity in a neighboring area, not where you have the grid cells, but in the area next to it, where activity actually reflected how much time had passed. This was seen in some individual cells. So the one that has is shown in the red box shows activity as a function of time. So time is on the x-axis. On the y-axis, you have the firing rate. That means the activity of that particular cell. And then you can see that the activity goes up and then down, up, down, up, down. So this ramp of activity occurs each time the rat is in the box for about 10 minutes. then it goes out and it goes to a new box and it begins slow again and then ramps up until the end, and then does this each time the rat is in a box. So this is a total then of about 10 boxes. So certainly there was some activity that reflected time, and you could also find the fractional cells where time actually explained much of the activity, which then in this brain area, we call it LEC, that's the lateral entorhinal cortex. It's not important for this context. But it's a unique brain area where activity of the cells actually reflects the passage of time. And that can be shown in, if you plot the activity, you break it down to the primary dimensions using a principal component analysis, and then you have the first and the second component here, and you plot the population activity. So each dot here is the activity at one given point of time among hundreds or even thousands of cells recorded at the same time, just as in the point cloud you saw with the torus. But now color shows the passage of time. So you can see that over time in this brain area, the activity actually it's not like on the torus where it moves on the same surface all the time, but it actually evolves and goes to different places all the time. So you can imagine in this high-dimensional space, it's like a snake that just moves around and finds new trajectories all the time. So this is a reflection of what we think is going on as the brain expresses the passage of time. And this just shows that you can, based on the activity of these many neurons, you can actually decode the point of time when a recording happened. So let me then just say that how does this come together? Where you have space, you have time. They are in slightly different areas of the entorhinal cortex, but come together in the hippocampus, and we believe these are essential and for forming the memories for which hippocampus is so important. So then for the final part of my talk, I want to try to connect it more to what you others are doing. So there are at least three major applications of neuroscience. And the first one is a very important one. I'm not going to talk about that today, but there are obvious health implications, right? Because psychiatry, neurology, there are so many syndromes that depend on the cortex and where we don't understand the activity that underlies these diseases. But I will also focus the rest of the talk on two applications. So first, briefly, on how you can actually make brains communicate with machines and vice versa. And then I will go over to the relationship or how neuroscience actually can interface with artificial intelligence. So let's begin with the brain-machine interfaces then. So I've just shown examples here. So this is how can brains and machines communicate. They both use electrical signals, so it shouldn't be a problem in principle to actually have them talk to each other at all. You can place electrodes just like we had in the mice or rats that were running around, that then pick up the electrical activity, and this can be fed into a computer or the other way around. So this is not a new invention at all. It has been going on for quite a while. And an example, the most familiar maybe, is the one shown at the bottom here, where people who, tetraplegics, who have lost sensation in both arms and in feet and can't move, can't sense anything, actually are able, based on their brain activity, to control the activity of a robotic arm, for example. So in this case, this patient has electrodes implanted in her brain. A computer interprets the signals. This is then obviously from the motor cortex, the part of brain that controls the movements. And then this goes to the computer, and the computer then moves that arm based on the neural activity in this brain area of the patient. Similarly, we have cochlear implants in the ears, which can then send signals to the auditory nerves. And there's also more, at a much more preliminary state, there's work on artificial vision, where you can try to send signals, visual signals, into there are several ways of doing it, but the most dramatic would be to send it directly into the visual cortex and actually stimulate cells inside the brain in cases where the blindness is, this is in blind persons, by the way, where the blindness is caused by, for example, degeneration in the eyes. So that doesn't quite work, but there is very promising work going on in that, and you can then imagine that there is a lot of potential here. I'll just show one example that was published in Nature in May this year. Again, a patient who could not move his arms and could not write, but what this group of people did was that they recorded activity from the cortex and then interpreted the activity, used machine learning approaches to distinguish clusters of activity depending on which letter this person was thinking about. For example, the person thought about the letter A or thought about the letter B or letter C and so on, and then the computer interpreted the electrical activity and then wrote the letter so that the person could actually write on a screen just by thinking about the letters. So finally then, I want to just try to relate a little bit better to artificial intelligence. So and of course, what is the task? Well, we all want to understand and perhaps even replicate or actually at least produce the functions that are so unique to the human brain, which human brain is good at, is to evaluate data that come in, sensory data that are very, very noisy, or many things that happen in parallel. Those are things that until recently at least computers weren't so good at. What is the potential for actually learning from the brain? Can AI learn anything from the brain? And I think the answer is both yes and no, but certainly there's a yes component. But there are some differences, and some of them are totally obvious, namely that the hardware is different, of course. In the brain there are neurons, but these neurons operate in circuits, so I will come back to that. But those circuits have many things in common with how computers work, because it is just at the more abstract level than the neurons. But there are also major differences that we don't understand, such as how can the brain operate at just 20 watt, whereas the supercomputers that perform only a tiny fraction of what the brain is doing still operates at needs many megawatts. So what is the trick that the brain uses to be so efficient? Another major difference: neurons are very slow. An action potential, which is the unit of communication, is about 1 millisecond, and the action potentials are the result of integration of activity that occurs before that, and they also give rise on the other side of the synapse to potentials that are much, much slower, up to 20 milliseconds. So this is very, very slow, but still the brain can be extremely accurate, and of course that is because you combine the activity of many neurons. Then I mentioned the binary nature of communication in form of action potentials, but then in addition the brain also does a lot of analog communication. So the synaptic potentials, as we call them, they are the ones that are between the action potentials, which integrate activity from many inputs. Those occur at a much slower scale, and they are analog. They can have any kind of value. And finally, a major difference also: brain operates probabilistically, so you can never really tell that a neuron is going to do this or that, because there's also always something that may happen. It's just a high probability, but we never know really. So, but similarities, certainly that the way brains work is that they work in networks, in circuits of neurons that work together, and as far as I understand, have some principles of organization similar to that what may be used in some computers as well. And the other thing is the ability to learn, which then brings me to machine learning, which has the ability to learn and modify one's own performance based on experience is certainly what makes the brain unique, but also what has made such advances in AI during the recent times. So in the brain, again, what you see is the synapses, the connections between neurons, and they can be modified all the time. And this is really what makes the human and the animal brain so efficient in adapting to the circumstances. So bottom line then, I think that there are important things to learn from each other. It occurs mostly at the algorithmic level in neural circuits, and where computational neuroscience might have something to offer AI. But it goes both ways, and this is my last slide then. I just want to take one example here. So what you see here is the use of AI to actually decipher what would be the best code for space. So this is in a computer, it's not in a biological organism at all. But what they did here was to provide this computer with information about direction and location. And the network by itself then developed grid-like units out of no instructions about being grid-like at all. So it actually shows that this could actually be an efficient, there's some advantage of doing it this way. And then they implemented this in a robot, then moved around in an environment, and actually in VR. And the nice thing is that this robot could then, or this machine could then navigate in this environment, take shortcuts, find more smarter ways to go from one place to the other. Then, and that was especially the case if they used this grid cell code, and not so much the case if they used other ways to represent location. And the good thing here is also that this computer actually outperformed a human game expert who's very skilled in moving around in environments in computer games. Just shows that actually AI can also be used as a tool for neuroscientists. It's not, it actually can go both ways. So with that, I think I finished my talk, and then I just would like to say that there are many people behind. I put the names here, and many people have paid for it. And then I think with that, I just thank you all for your attention.